Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and a very good afternoon to our guests. Yang berbahagia, Prof. Rafiza Haji Muhammad Ismail, President of Malaysian Society of Pharmacology and Physiology. Yang berusaha, Associate Professor Dr. Muhammad Farouk Sheikh, Chairperson of MSPP 34th Scientific Meeting 2021. Yang berusaha, Dr. Muhammad Helmi Mokhtar, Chairperson of MSPP Young Investigator Award 2021 Organizing Committee. Professors, doctors, ladies and gentlemen, it's my pleasure to welcome all of you here this afternoon to MSPP Young Investigator Award 2021 competition. I'm Dr. Adila, will be the MC for today's event. Before we begin, let's open the session by reciting Surah Al-Fatiha. Ladies and gentlemen, we have three shortlisted candidates competing for MSPP Young Investigator Award competition this year. They are Dr. Azizah Ugusman from Universiti Kebangsaan Malaysia, Dr. Tan Chu Hock from University of Malaya, and Dr. Asthma Abdullah from University Science Malaysia. All the contestants have met the criteria, which are MSPP members aged 40 or younger on 31st December 2021, conducting research in any area of physiology or pharmacology, and currently resided and conducting research in Malaysia. Before we start with the presentations, allow me to introduce the panel of judges. We are honored to have three judges with us today. Our first judge is Prof. Dr. Nafiza Haji Muhammad Ismail, Immediate Past Dean, School of Medicine, International Medical University. Next, we have Prof. Igor Nikolayevich Lizitsa, Professor of Pharmacology, School of Medicine, International Medical University. And last but not least, we have Prof. Dr. Uma Devi M. Palanisamy, Professor of Biochemistry at the Jeffrey Chia School of Medicine and Health Sciences, Monash University. Before we start, let me brief you about the rules and timings of the competition. The speaking order of the contestant is: the first one is Dr. Asma Abdullah, followed by Dr. Tan Chu Hock, and the last one is Dr. Azizah Ugusman. The contestants' pre-recorded 10-minute video will be played first, followed by a live questions and answers session for 10 minutes. And after the contestant has finished his or her session, there will be a 2-minute interval to enable the judges to give their scores. Okay, due to the time limitation, we do not allow questions from the attendees. However, if you have any questions, just type at the chat box and maybe the respective presenter can answer at the chat box. So the scoring sheets, I believe, have been sent to the judges via email. All right, so we will begin the presentation. Ladies and gentlemen, let's get the ball rolling with the first finalist, Dr. Asma. Associate Professor Dr. Asma Abdullah is a lecturer in School of Health Sciences, USM. She graduated with a PhD in molecular immunology from USM in 2008, and her research interests are molecular immunology, regenerative medicine, and vaccinology. We will now listen to her 10-minute presentation recording.
Assalamualaikum warahmatullahi wabarakatuh and salam sejahtera to the honorable professors, doctors and panel of judges. My name is Nura Asma Abdullah. I'm from School of Health Sciences, University Science Malaysia. The topic of my presentation today is anti-asthmatic effects of Rhinoceros rhinocerotis, a potential alternative for allergy asthma management. Asthma is defined as a chronic disease of the airways, which involves inflammation and narrowing of the airways, and this making the sufferers difficult to breathe. At the moment, asthma is incurable. However, with proper medication, it can be managed. So the inflammation of the airways causes recurrent episodes of symptoms such as wheezing, breathlessness, cough, and also chest tightness. So in the chronic asthma, so other clinical manifestations such as airway hyperresponsiveness, eosinophilic lymphocytic inflammation, as well as remodeling of the airway walls. So in the pathophysiology of asthma, so the common characteristic is characterized by the thickening of the airway wall. So the thickening of the airway wall resulted in the narrowing of the airway lumen. So during the asthmatic attacks, the smooth muscle become tightened and makes the lumen become narrower, and this makes the asthmatic patient difficult to breathe. So in brief, on the pathogenesis of allergy asthma or classical asthma, so it is induced by the presence of allergens. So these allergens, when it comes close to the epithelial of the airway, it will be taken up by a specific cells called dendritic cells. So these dendritic cells will process and present these allergens to T lymphocytes of class 1 and class 2. In a relative expression of class 2 T lymphocytes, or we call it FTH2, there will be two consequences. The first one is with the release of interleukin 5, there will be activation of eosinophil, which produce more cytokines and leukotrienes. And the second consequences, the activation of B cells, in this figure is plasma cells, which is mediated by type 2 cytokines, Th2 cytokines, interleukin 4, 5, and 13, to produce an antibody called IgE, immunoglobulin E. So this IgE will bind to mast cells, which then release the mediators called histamine, leukotrienes, and prostaglandin D2. So these events contribute to the pathophysiology of allergy asthma. So the common pharmacologic therapies for asthma is defined by two approaches. The first one using bronchodilator therapy, and the second one is using anti-inflammatory therapy. So these medications are either taken as controller or relievers, and most of the time asthma patient using it in combination. So this medication usually come with various side effects when taken for a long period of time. So to reduce dependency of the asthmatic patients to this medication, an alternative to the current medication to reduce dependency to the current medication is needed. So I would like to introduce our focus of study, Dinosus rhinocerotis, is a medicinal mushroom which available in the forests of Southeast Asia. So this Dinosus rhinocerotis is commonly used by the orang asli population in Malaysia to treat their chronic cough and also asthma. So since 2012, we have been investigating these Dinosus rhinocerotis extracts in allergy asthma model. So in this study, we are using two types of allergy asthma models. The first one is ovalbumin-induced model, and the second one is house dust-mite-induced models. So in this presentation, I will share some data that we generated from these two models. So this slide shows the effects of LRE on Th2 cytokines level in the bath bronchoalveolar lavage of ovalbumin-induced mice. So LRE reduced the level of interleukin 4, 5, and interleukin 13 in ovalbumin-induced mice. So we also investigated the effects of LRE on inflammatory cells, and this figure we showed that LRE significantly reduced the level of eosinophil, neutrophil, and lymphocytes in the ovalbumin-induced mice. We know that these inflammatory cells contributed in the pathogenesis of allergy asthma. So it's interesting to show here that the treatment of LRE also modulates the level, the population of Tregs in the ovalbumin-induced mice. So with the treatment of LRE and also dexamethasone, there were a significant increase of the Treg population in the OVA-induced mice. So we also showed that LRE reduced the level of IgE in the serum of an OVA-induced mice. So we also investigated the effect of LRE on the airway hyperresponsiveness. So with the HDM induction, we demonstrated significant increase of the airway resistance. Interestingly, when LRE and dexamethasone administered respectively, so there was significant suppression of the airway resistance in the HDM-induced mice. So we also investigated the effect of LRE on histological parameters in OVA-induced mice. So this figure showed that LRE significantly suppressed the number of leukocytes infiltration in the OVA-induced mice. So allergy asthma also is characterized by the accumulation of mucus in the lung tissue. So we also measured the effect of LRE on goblet cell hyperplasia. So LRE significantly suppressed goblet cell hyperplasia in ovalbumin-induced mice. So we also demonstrated the effects of LRE on airway smooth muscle thinness. And this figure shows that the LRE significantly reduced airway smooth muscle thickness in ovalbumin-induced mice. There are several studies reported the involvement of TGF-beta in the allergy asthma, specifically in the tissue repair and the remodeling of the lung tissue. So in the LRE treated group, we demonstrated significant reduction of the TGF-beta positive cells in the OVA-induced mice. So we also investigated the effects of LRE on activin A, a cytokine which play immunoregulatory roles in allergy asthma. So we showed that with LRE treatment, LRE significantly reduced the number of activin A positive cells in OVA-induced mice. So it's interesting to note the involvement of LRE in molecular mechanism of allergy asthma. So we investigated further to check the effect of LRE on the key regulators in allergy asthma. So with ovalbumin induction, there were significant upregulation of the key regulators in asthma. Interestingly, with the LRE treatment, these genes were downregulated. So it gives us interesting direction to explore further the involvement of LRE in the molecular mechanism of allergy asthma. So as a conclusion, LRE demonstrates anti-asthmatic effects by ameliorating the airway inflammation, hyperresponsiveness, and remodeling, thus suggesting its therapeutic effect, therapeutic potential as an alternative for the management of allergy asthma. I would like to thank these researchers for their contribution. Thank you very much for listening to my presentation today. Thank you.
Thank you, Dr. Asma, for the presentation. I now open the floor for questions from the judges.
Can I go first?
Yes, please.
Okay. Hi, Asma. It's a very interesting presentation, and it looks like LRE seems to have very good anti-asthmatic effect. I just have a couple of questions. Why did you choose those dosage levels, 125 to 500 mg? And all my questions are connected, so I'll just tell you all of them. Why did you choose that dosage? Was toxicity studies done at that dosage? And it looks as though you use a crude extract from your presentation. Has anyone identified the compounds, or have you guys identified active compounds from this mushroom? Yeah.
Okay. Thank you, Prof. Umar, for the interesting questions. Okay, I would like to ask the first question. Okay, the concentration that were chosen in the study is actually based on the based on the toxicity study that were performed by one of the a group that actively working on this Diagnosis rhinocerotis in Okay, so they will actually tested the toxicity level of this mushroom up to 2000 mg. So within the range, this concentration basically safe to be administered in the animals. So that's and based on our preliminary studies, so this the range of concentration that gave a good effects in the animal model. Okay, and the second question is basically on the bioactive components, right? Okay, so there are quite a number of studies reported because this Lichnosus rhinocerotis is a medicinal mushroom. Okay, so it's not a plant, so it's a medicinal mushroom. So there are a number of reports demonstrated the main composition in the medicinal mushroom mostly is the carbohydrates composition. So in this case, we are referring to the glucan. So there are a number of studies that reported the effects of these carbohydrates fractions, or some actually reported the glucans fractions in their animal study. So our group now is currently investigating the effects of polysaccharide-based fraction in this allergy asthma model. So it's interesting result that we will share soon, hopefully, with all the researchers. So that's my answer to your question.
Okay, thank you.
I've got a couple of questions. First of all, congratulations, Asthma. I thought your study was very... Can you hear me?
Yes, clearly.
I thought your study was very interesting, showed interesting findings, especially on the genes, right?
Thank you, Prof. Nafisa.
Can I ask why you chose dexamethasone as your corticosteroid control? That's number one. Number two, can you summarize the difference in the findings between the mushroom and DEXA on the hyperresponsiveness of the airway?
Okay, thank you very much for the question. Okay, so in this study, we have chosen dexamethasone because it works as anti-inflammatory drugs. Okay, so this is a common drugs that actually use in the management of inflammation. So in our airway hyperresponsiveness studies, it's very interesting to note that the results from LRE treated groups in any concentration basically give a comparable effects with the dexamethasone. However, we know that we need a further study to actually look at the mechanism, okay, the mechanism, and also on the bioactive composition of this mushroom, basically, which basically triggering the effects.
Thank you. May I suggest? I'm sure you're going to continue the study.
Yes, definitely.
To use corticosteroid, which is actually used in asthma to treat asthma in patients, and dexamethasone is not used. All right, so maybe
yes,
so it's more akin to reality.
Yeah. But we actually referring to the previous studies, and dexamethasone is commonly used in the animal models for allergy asthma. So I will take note on your suggestion, Prof. Definitely.
And one last thing, in your abstract, you had written that it can be used as an adjuvant.
Yes.
Therapy, but you concluded in your last line that it's used as an alternative. So which is it you're recommending?
So basically, what we are proposing for this Lichnosus rhinocerotis is actually to be taken as an addition to the current medication for allergy asthma. So yeah, so that's what my answer to that.
So it's an adjuvant and not an alternative.
Yeah, sorry. Maybe, yeah, the correct word is adjuvant.
Thank you.
Mhm.
If I may ask a question.
Yes, Prof. Igor.
Dr. Nurul Asma, first of all, my congratulations. Very interesting work. And
Thank
I
you.
really enjoy listening to your presentation. I have a couple questions. The first question, I think Prof. Nafiz already highlighted, the reference drug which you used. It's one of the most potent anti-inflammatory drugs, dexamethasone. And your compound was like comparable with the effects, with anti-inflammatory effects of dexamethasone, right? Any suggestion, what could be the mechanism of action? What could be the possible mechanism of anti-inflammatory effect of your compound? The first question. And another question, a part of anti-inflammatory properties of lignous rhinocerotis, right? So are any other pharmacological properties were studied before? Maybe bronchodilatory effect or mucolytic effect of this compound, which could be additionally have some benefits in the treatment of the bronchial asthma?
Okay. Thank you very much, Prof. Igor. So, so to answer your first questions on what's the anti-inflammatory mechanisms of this Linosis rhinocerotis. So we are actually looking forward to the mechanism of study. However, I mean the comment, because we have not come to the molecular mechanisms of in details. However, if you can see that this Linosis rhinocerotis extract also has some, effects to the TGF-beta as well as activin A. So it can be maybe part of the molecular mechanisms of this LRE, this Linosis rhinocerotis extract. And our next study is basically to look at the, because we are now working on the crude extracts. So the next study, when we work on the polysaccharide fractions, we would like to explore further on the molecular mechanisms of some growth factors, such as that actually involved in the inflammation process. So hopefully in the future study, we can explore further on the effects of these extracts on the prostaglandin pathway, or maybe on histamine pathway. So that's our next directions in this study.
Okay, yeah. And the second question was any other pharmacological effect apart of the anti-inflammatory properties, such as bronchodilatory effect or mucolytic effects are known for this compound?
Okay, so we have done some preliminary works on the receptor function. So that one is also our aim in this study, is to look at the effects of these extracts on the receptor function. So I hope I cannot answer your questions for now because we have not shared the information in these slides. So we hope that we are now working towards that.
Okay, thank you. Thank you.
Okay, thank you, judges. We will now have 2 minutes for the judges to give their marks. I think there's question from the attendees to Dr. Nur Asma. So Dr. Nur Asma can type the answers in the chat box. All right, okay, next. It's my pleasure to introduce the second finalist, Dr. Tan Chu Hock. Dr. Tan Chu Hock graduated with a PhD in pharmacology and toxicology from Universiti of Malaya in 2013. He is currently a lecturer in Department of Pharmacology, Faculty of Medicine, UM. His research focuses on the pharmacokinetic and pharmacodynamic aspects of venom-antivenom interplay. Yeah, Dr. Shafi, can you please share the slide? So to know more about this, we'll listen to his 10 minutes presentation recording. Dr. Shafi, okay, thank you.
Good afternoon. I would like to share with you my presentation entitled Where Pharmacology and Venomics Meet Towards a Comprehensive Solution for Snakebite Envenomation, a Priority Neglected Tropical Disease. Now, snakebite envenomation kills more than 100,000 people every year, with 4 times as many continue to suffer long-term complications. Indeed, it is perhaps the most important tropical disease you have ever heard of. This is well recognized by the World Health Organization, who also highlights that effective treatment with antivenom is the crucial solution. However, venomous snakes are highly diverse and they are widely distributed. Yet, the species coverage and supply of antivenom remains very limited in many parts of the world. So, looking at the problems, we hypothesize that snake venom compositions are variable at both inter and intraspecies levels, and the variation can result in differential toxicity and antigenicity of the venom. Hence, deciphering the variation is important to understand the pathophysiology and to improve the antivenom treatment. So we designed a series of studies that first look at the biochemical composition of various Cobra venoms through proteomics and transcriptomics approaches. This is followed by toxicity study of the venoms and their respective toxins, and the neutralization activities of various antivenoms against these venoms and their toxins. And finally, we explore the development of a new antivenom which carries the hope to overcome the shortcomings of existing antivenom products. Most of you can recognize that this is a cobra. But bear in mind that cobras are more diverse than this, and they are widely distributed into many different geographical populations. My venomic study have revealed substantial venom variations between and within the different species of cobras, but also identify 2 common groups of key components in the venoms, which are essentially post-synaptic blockers called the short and long chain alpha neurotoxins. Now, these neurotoxins have very low LD50, which means they are very potent neurotoxins. With that, we established that the relative abundances of alpha neurotoxins in the venoms are well correlated with the venom lethality, and this explains the consequence of venom variation on the differential toxicity shown by the different cobra species. Then, we evaluated the neutralization activities of the various antivenoms for Cobra venoms in order to understand the impact of venom variation on the neutralization effectiveness of these antivenoms. In the nerve muscle preparation, immunocomplexation study shows that, for instance, Thai cobra, which is a Niger-Calvia, venom requires 4 times higher dose of antivenom to prevent paralysis. Now, this is actually consistent with the much higher abundance of the alpha neurotoxins in its venom. Now, in vivo study, the efficacy of antivenom is again low in the case of venoms which contain abundant alpha neurotoxins. This highlights the fact that with a higher abundance of alpha neurotoxins, the antivenom is likely going to be less potent, and clinically this means a higher dose of antivenom will be required. This unfortunately will predispose the patients to a higher risk of hypersensitivity reactions, which is equally fatal, and it also increases the treatment costs. We also examine the effect of time of treatment on the reversal of muscle paralysis caused by the Cobra venoms. Consistently, we observe that when the antivenom is given at the later cause of the paralysis, it became more difficult to restore the neuromuscular functionality. Now, this infers that in Cobra bite envenomation, antivenom treatment should be commenced as early as possible for an improved prognosis and a better clinical outcome. In experimental envenomation, or what we call a challenge rescue model developed in my lab, we noticed that when the mice is challenged with Cobra venoms, which are invariably containing a higher abundance of alpha neurotoxins, it is associated with a rapid onset neurotoxicity. Now, it is possible to prevent death with the use of antivenom, but typically it requires a higher dose of antivenoms and a longer time for full recovery. So this highlights the limitations of the various antivenoms against Cobra envenomation. And we further expanded our study to also look at the various other highly neurotoxic Elapid snakes in the region in order to identify the distinct key neurotoxins and also the impact of venom variation on their neutralization. So with the existing data that we've got and also the Venomics protocol developed in my lab, we were able to identify, to purify and characterize the various species-specific key toxins from all these species. And this actually opens the doors for various applications which my group is currently happily working on, including works like diagnostics, drug discovery, and most importantly, which I'm going to share with you, is the development of a potentially universal antivenom for improved Cobra bite treatment. So with our findings, we came up with a principle that antivenom should target the right toxins of the right species tailored to its locality. So we innovated an immunogen mixture, which then is used to raise horse antiserum. And we showed that it was able to cross-neutralize more than 30 neurotoxic venoms from virtually across the globe. So this actually is promising because it sort of addressed the existing problem of the limitation of many antivenoms in terms of species and geographical coverage. And my team is currently further looking into possible refinement of such universal antivenom through optimization of the antigen formulation, the immunizing techniques, and also methods for antibody purification. So in conclusion, by integrating Venomics and pharmacology, we were able to gain deeper insights into the venom diversity, which comes with medical implications. Now, it is important to understand the venom composition variation and the ensuing functional variances, because this will allow us to elucidate the pathophysiology of snakebite envenomation, and importantly, to pave the way for the development of a pan-species, pan-regional antivenom to save more lives. So I would like to acknowledge the following organizations for their support and collaborations. And on behalf of my team, I would like to thank you very much for your time and attention.
Thank you, Dr. Tan Chu Hock, for the presentation. So now I open the floor for questions from the judges. So I see Prof. Nafiza. You want to start first?
Yeah, okay. Thank you, Dr. Adila. Dr. Tan, very impressive research and interesting findings. And I agree with you, it's underexplored. And I'm glad you're having a lab at Pharmacology UM to explore this area. My question to you is, when a snake bite patient arrives at ED of UMMC, do they consult you? That's number one. Number two, if they have not consulted you, which means it's not ready yet to be administered to patients, how long do you think it will take for it to be applied to patients? Because that's what we want to do, right? To improve the quality of life of people.
Yes, Prof. Thank you for the questions. When patient was admitted to the hospital, usually the first thing the doctor would want to know, of course, after stabilizing the patient, is to get the diagnosis of the snake. So in terms of the diagnosis part, at the moment, there is no diagnostic kit available, although my group is also working on that. So what they do is usually they will get the photo of the snakes. Nowadays, we advise the patients to actually take photos instead of catching the snake, to prevent, of course, secondary bite and so on. And then sometimes the doctors or even the paramedics or the patients themselves, they would have actually sent the photos to us, either to our group or there's also another team, clinical team, which we are also sort of working together with, called the REX. Consultation service. So usually we will be able to identify the snakes and provide the information from there on about the choice of antivenoms and whether it is necessary or whether it is indicated in those situations. As for how long it will take, it depends on the species, of course. That's why my project is, we are currently looking at the rapid onset neurotoxic snakes. Typically, like a cobras and so on, because of the huge amount of venoms and also the very fast action of these alpha neurotoxins, the paralysis can set in within hours. So of course, the earlier you could give the antivenoms, that would be the better, give you a better prognosis. I hope I answer your question, Prof.
Yeah, I think it's, I understand that it's very difficult to stipulate a timeline.
Yes.
Yeah, but I was just thinking whether you are on track with some of the venoms. But anyway, thank you, Dr. Tan, for your answer.
Okay. If I may ask question, Dr. Tan?
Yes, sure, Prof.
Okay, I have few questions. The first of all, I would like to congratulate with your work. It's really interesting presentation. I enjoy very much. And okay, the first my questions, are there any variations in the venom content within the same species when they was taken from the different part of Southeast Asia? For example, for the Naja cotia or the Naja sumatrana, if they're taken from the different part of Southeast Asia, any variation in the venom content is the first question. Okay, how many samples did you have? I would say, how many snakes from the same species was collected? Second question. And the third question, also it is well-known fact that there is variations in the content of the venom within the same species depend of the age of the snake and the many other factors. So how basically you avoid this bias in your research?
Prof, would you mind to sort of repeat the third question?
Third question, I mean that the content of venom within
Yes.
the snake, same species could vary, isn't it?
Yes.
It could depend on the age, I mean how old the snake, it could be seasonal season
Yes.
variations,
Yes.
right?
Yes.
So I mean, so how you avoid this kind of the variation in your study?
Okay, thank you, Prof. To answer the first question about the variation in terms of geographical, contacts, it is well defined, already established, and in fact, my group is one of the first to actually look into the geographical venom variation in, for example, Naja cotia, that is, prevalent in our in Southeast Asia. In fact, one of the example that I highlighted in the slide just now, unfortunately, the slides, it doesn't really appear smoothly just now. But actually, we are trying to highlight to you that with geographical variations, actually the same species, even though they are from the same species, but different geographical populations may have variable venom compositions. And because of this, it comes with different protein antigenicity that will inevitably impact the immunorecognition of the antivenom available. Like the Naja cotia antivenom we have now is all from Thailand. And so it will exhibit differential effectiveness or efficacy against the same species venom, but sourced from different geographical area. And so we actually look into these problems, and as far as we can, this is also in line with the WHO guideline, is that whenever you try to address the efficacy of an antivenom, you should look into the variations. And this also connect to Prof, your third questions about the other factors of venom geographic, the venom variation, which includes the ontogenic, like the different development of the snake, whether they are juvenile, whether they are newborn, they are adult, and the seasonal. Of course, Southeast Asia seasonal maybe not so affected, but yes, we do take into those consideration, and we actually also look into the sexual dimorphism because the different sex and so on, you also can exhibit venom variation. So in our study, where it is possible, we will try to get the samples along with our helps from herpetologists and so on to get as many specimens. So easily we will try to make a minimum of at least 3 to 5, but we cannot deny the fact that it is not easy to get the specimen. And usually I will have to be there to authenticate the species, and then we have to milk the venom ourselves and so on. So we will try our best to cover the different spectrums, like different ages of the snake, different geographical and different sex and so on, as much as possible, so that we can not to say avoid the variation, but we take them into consideration, so that when we do the venom profiling, we can see the differences very clearly. And also when we check the immunogenicity, the epitopes and so on, we are sure, whether there is venom variation and whether they respond differently to the antivenoms available. Yeah. So I hope I answered your question, Prof.
Thank you very much.
Hi, Dr. Tan.
Prof.
Definitely agree that your work is very pertinent, particularly in the Southeast Asia region, where the development of antivenom is not as progressive, other countries. I have two clarification and one question. So my first clarification is that I believe your study is kind of a multi-center study where you notice that different countries were doing.
Definitely.
Yeah. So did you use this, did all of these countries use the same efficacy studies? Because when you presented, you showed that your efficacy studies and you compared it. So did you all use the same type of study?
Okay. Yeah, thank you, Prof. So in this project, actually, we collaborated with, multinational, because we are not looking at only Malaysia, but Southeast Asia. In fact, we also, collaborated with people from China, Taiwan, and then even Sri Lanka and India and Pakistan as well. So I think maybe we are fortunate that my lab is equipped with the efficacy. In fact, we call it the pharmacology testing, the classical and also the more, the kind of molecular kind of work. So so far, most of the efficacy testing were done in our group.
Okay.
So we are adopting the same, the standard protocols according to what is, of course, WHO has a guideline as well. But of course, we also modify that. I mean, as a because I was in the duty of experts there. So we actually also sort of like improvise or improve the methods from time to time.
Okay.
So I think at the moment we are quite fortunate because we are able to do the venom and also the anti-venom testing using a standardized way, a standard way, protocol that is available in my lab. Yeah.
Okay. So the second clarification, you mentioned that the time of anti-venom therapy was very important. So was this finding across all varieties?
Yes, in a way, of course, the different effects like neurotoxic or hemotoxic, for example, they may respond differently. But as what our universal antivenom that we are developing currently is aiming at, is we hope to actually arrest the neurotoxic, the paralysis effect, which is the main killer, So yes, the time is important in such a way that the early you give the antivenom, then it seems that it is much easier to arrest the neuroparalysis and also to restore the muscle contractions. But it differs with the venom variation, as what you have mentioned. The different species, they may contain different proportions of snake venom, the neurotoxin, for example, the alpha neurotoxins. So the more the alpha neurotoxin is, then it will be much difficult if you are giving the antivenom at a later cost. So this venom variation is not just occur interspecies, but also intraspecies, as what I have answered Prof. Igo just now. And so this is, I think it is an important issue when we design the time of treatment for antivenom. It has to be tailored to the species. Any intraspecies variation, and just if I may share with you, I was talking about alpha neurotoxin, which is a post-synaptic neurotoxins. I think this morning, Prof. Win also talked about that. But there is also another group of pre-synaptic venom, which is actually they act by destroying the nerve termini. So in those cases, once the nerve terminals are destroyed, then even though you give the antivenom, it won't be able to regenerate in time. So therefore, in those pre-synaptic neurotoxic envenomation, you need an even earlier antivenom administration. Even now, people are debating, whether you should give it prophylactically, before the patients start developing ptosis and weakness, things like that. Yeah.
My last question, Dr. Tan, is that your suggestion of developing, or idea of developing a universal antivenom, I think it's very
thank you.
Very good. But do you foresee any issues with it? What are the disadvantages do you think would be if you have a universal antivenom? Potential disadvantages, yeah.
The advantage and challenges. Okay. The advantage, of course, is we can address the main two issues about the current antivenom limitations. One is the species specificity. Different species and even the same species, they have venom variation. So you need a very, very, very specific antivenom usually. So that's the first thing about the spectrum of species coverage. And the second issue is the efficacy. So our proposed universal antivenom, in fact, is to address the two issues. And therefore, by improving the species coverage, which I have shared just now, that we are able to cross-neutralize basically the 4 continents neurotoxic venoms, that would address the species coverage. So it will have a wider geographical utility. And secondly, the efficacy, which we are now optimizing, is to try to, from the anti-serum, then how we are going to improve the, the purification to increase the immunological binding activity and so on. So this is the strength or the advantage. But as for the challenges, I think is the main challenges that we are facing is the research interest that we can get, because we foresee that going down, we may need to attract pharmaceutical companies to look into this. But as you know, this is a rather neglected, problems, and antivenom is an orphan drug, and it's not going to make much because the end users are those in the poor countries and so on. But we hope that, with the rising awareness and also collaboration from WHO, who looks into this currently, we are able to overcome these problems in terms of the pharmaceutical investment, the fundings, etc., etc.
Okay, thank you very much. Thank you, Prof. Umar. Thank you, Dr. Tan Johor. I think that's the only time left for question and answer. So shall we move to the next presenter? All right, our next, okay, so we have another 2 minutes for the judges to give their marks before we move on to the next presenter. Thank you, judges. Thank you, Dr. Tan, for the presentation again. And now let us welcome the final contestant, Dr. Azizah Ugusman. Dr. Azizah Ugusman is an associate professor in Department of Physiology, Universiti Kebangsaan Malaysia. She graduated in 2012 in UKM with a PhD in physiology. She is actively involved in cardiovascular physiology and pharmacology research, which includes study on perivascular adipose tissue and natural products for cardiovascular health. We will now listen to her 10-minute presentation recording.
Good afternoon, everyone. I'm Aziza from Physiology Department, Universiti Kebangsaan Malaysia. Thank you, MSPP, for shortlisting me for the Young Investigator Award this year. Today, I will share with you my research findings entitled Role of AMP-activated protein kinase in modulating perivascular adipose tissue function. Have you ever been caught in a situation where you frequently ordered food delivery service and resort to the couch potato lifestyle? And the next thing you know is your old pants can no longer fit, your body weight increases, and you end up becoming obese. If the answer is yes, you are certainly not alone. Do you know that worldwide obesity has nearly tripled since 1975? Most people live in countries where overweight and obesity kills more people than starvation does. In Malaysia alone, in 2019, 1 in 2 adults were overweight or obese, and unsurprisingly, Malaysia is also the fattest country in Southeast Asia. I believe with the current pandemic situation, with us being confined to our homes, this statistic will even be worse. Obesity is a risk factor for cardiovascular diseases, which are the main killer not just in Malaysia, but also worldwide. Obesity affects various fat depots in the body, including the fat surrounding the blood vessels known as perivascular adipose tissue. Perivascular adipose tissue, or PVAD, which is located at the outer part of the adventitial layer surrounding almost all blood vessels except for the cerebral vessels, is composed mainly of adipocytes. As an active endocrine and paracrine organ, PVAD secretes a range of vasoactive adipokines that actively communicate with the underlying vasculature to modulate vascular function. In pathological conditions such as obesity and cardiovascular diseases, the adipokine profile of PVET may be altered, a condition referred as PVET dysfunction. In healthy conditions, PVET produces more anti-contractile adipokine, such as adiponectin, to exert a net anti-contractile effect on the underlying vessel. However, pathological conditions, such as obesity, may alter the secretion pattern of PVET, leading to an imbalance in favor of more pro-contractile adipokines, thus causing vascular complications. However, it is unknown whether alterations in the PVET AMPK activity is the underlying mechanism of such effects. AMPK is a 3 subunits enzyme that consists of alpha, beta, and gamma subunits, but the catalytic unit is the alpha subunit. The alpha subunits exist in 2 isoforms, alpha 1 and alpha 2, with alpha 1 as the main isoform expressed in vascular tissues and PVET. Although AMPK is often described as a modulator of cellular metabolism, it also has important roles in maintaining vascular homeostasis. AMPK activation stimulates more nitric oxide production in the endothelium to cause vasorelaxation. AMPK can also induce endothelium-independent relaxation of the vascular smooth muscles by causing the opening of potassium channels. Although it is known that AMPK can modulate vascular smooth muscle cells and endothelial functions, it is unknown whether AMPK can influence the anti-contractile activity and adipokine profile of PVET. Despite AMPK activation having beneficial effects on endothelial and vascular smooth muscle functions, AMPK role in mediating the anti-contractile effect of PVET under basal and obese conditions is not well established, hence the need of this study. We hypothesize that activation of AMPK in the PVET could improve the PVET adipokine profile, which may then have beneficial effects to control the tone of the underlying blood vessel, and this mechanism may become deranged in obesity. Therefore, the aims of this study were to determine the role of AMPK in modulating the anti-contractile action of PVET, as well as to assess the effect of high-fat diet-induced obesity on the anti-contractile action of PVET and whether any changes involve AMPK. The anti-contractile action of PVET was studied using biomyography in endothelium denuded aortic rings of wild type and AMPK alpha-1 knockout mice either with or without PVET. We then contracted the vessels using U46619 followed by cumulative dose response curves to a vasodilator chromacalin. The results showed that the presence of PVET significantly augmented the relaxation to chromacalin in wild-type aortic rings. However, in aortic rings from AMPK alpha-1 knockout mice, the presence of PVET had no effect on relaxation to chromacalin, indicating that the PVET of AMPK alpha-1 knockout mice is dysfunctional. To clarify how AMPK affects the secretion of adipocytokines by PVET, we performed an adipocytokine array. And the most striking difference was a reduction in adiponectin in conditioned media prepared from knockout mice PVET. Quantitative studies using ELISA further confirmed a significant reduction in adiponectin in the knockout mice PVET conditioned media. Adiponectin effect on the vasculature is mediated via its binding to adiponectin receptor 1 or AdipoR1. Blocking adiponectin receptors in wild-type aortic rings with intact PVET significantly attenuated the relaxation to chromacalin, while in knockout arteries with intact PVET, the blocking peptide had no significant effect. Adhesion of exogenous adiponectin to wild-type aortic rings without PVET significantly augmented the relaxation to chromacalin. In knockout mice, incubation with adiponectin also significantly enhanced the relaxation to chromacalin in aortic rings without PVET. Taken together, these results further confirm that adiponectin is the pivot-derived relaxing factor and AMPK alpha-1 knockout mice releases less adiponectin. In the next stage of the study, we tested the effect of high-fat diet-induced obesity on the anticontractile action of pivot. The results show that obesity attenuated the relaxation to chromacalin in aortic rings with attached pivot, but not in the rings without pivot. To further clarify the effect of obesity on AMPK level and activity, analysis of the levels of total and phosphorylated or activated form of AMPK alpha in the pivot was performed. Pivot of obese mice showed a reduction in the levels and activity of AMPK alpha. And this is associated with reduced adiponectin release from the obese pivot samples. In summary, pivot from AMPK alpha-1 knockout mice exhibits reduced anticontractile activity due to a reduction in the secretion of adiponectin. AMPK alpha-1 plays a critical role in maintaining the anticontractile action of pivot, an effect independent of the endothelium but likely to be mediated through altered adiponectin secretion. And lastly, obesity is associated with reduced AMPK activity and adiponectin secretion by pivot, leading to the loss of pivot anticontractile effect. So, what is the relevance of this study? It is well established that obesity is an independent risk factor for cardiovascular diseases, and cardiovascular diseases are the #1 killer not just in Malaysia but also worldwide. Hence, studies concerning the pathophysiology of obesity-related cardiovascular complications are of utmost importance for better management of such diseases. AMPK may protect pivot and the underlying vasculature against deleterious changes of obesity. Since AMPK is important for normal pivot function, AMPK activation is a promising therapeutic target for the treatment of vascular complications associated with obesity. The results presented have been published in the following original research articles. And these projects are the continuation of the work presented. I'm currently working on the crosstalk between AMPK, pivot, and endothelium in obesity, the effect of nicotine on the anticontractile function of pivot, and pivot imaging in young patients with coronary artery disease. Just sharing some of the activities I was involved in communicating science to the public and promoting the importance of cardiovascular health to the secondary school students. I would like to thank Prof. Simon Kennedy and the rest of the team members in University of Glasgow and Physiology Department UKM for their valuable input throughout the study. Thank you for your attention.
Thank you, Dr. Aziza, for your presentation. I now open the floor for questions from the judges.
I'll go first.
Yes, please, Prof.
Yeah, thank you, Aziza.
Thank you, Prof.
I thought it was a very, very clear presentation and very important presentation. My question is very simple. You mentioned that 1 in 2 people are obese and that obesity is an independent risk factor and for cardiovascular disease, and you're right. But my question to you is it overall obesity or abdominal obesity?
Okay, research show that abdominal obesity possess higher risk for someone to develop cardiovascular diseases than just overall obesity.
So what's the, you said 1 in 2 adults have overall obesity, either it's overweight or obesity, you plus a 2 and you get more than 50%, right? And what about abdominal obesity?
Yeah, for abdominal obesity, the prevalence is also 1 in 2 adults in Malaysia, as for 2019 NHMS.
What in your reading, not in your research, in your reading, how is this related to diabetes?
Okay, when someone is having abdominal obesity, they are at higher risk to develop insulin resistance and diabetes because of altered insulin sensitivity at the skeletal muscles, and therefore someone with abdominal obesity has higher risk to develop diabetes as part of the metabolic syndrome.
Thank you. No more questions.
Thank you, Prof. Nafiza. Is there any other questions from other judges?
Yeah, I'll go. Hi, Aziza. Yeah, it's quite an interesting work that you've done. Just a few clarifications as well. I just did a quick search, and there was also a very similar. It's just a title, but I want to know how different is your study from a thesis presented in 2017, also from the University of Glasgow. It's very similar and almost exactly the same topic. So I believe you're also from there, but what was the difference in your study from that?
Okay, I believe the thesis was from my lab member, Dr. Tariq Al-Mabruk. Yeah, because this study was part of my postdoc research study. There are some parts of the work that belong to my project, for my postdoc project, and some parts of the work belong to Dr. Tariq Al-Mabruk's project. So for my study, I concentrated more on the myography work, while he concentrated more on the immunohistochemistry work.
Okay, so it's similar from the similar group now.
Yeah.
Okay. Now, how do you think your study, your findings that you have obtained regarding the AMPK involvement in obesity, what is its bigger picture in the management of, you talked about obesity, diabetes, and metabolic syndrome. How do you foresee its presence or in its prospective clinical management?
Okay, all right. For now, the mainstay management for obesity is lifestyle modification, okay, which consists of diet and also exercise. So in terms of this study, we showed that in healthy condition, in baseline condition, AMPK is important to maintain the pivot function through the secretion of adiponectin, which is one of the important adipokines secreted by pivot. And in obesity, AMPK function is inhibited, and thereby causing the adiponectin release to be reduced and pivot function to be altered. So from there, it is interesting to investigate whether the activation of AMPK can be beneficial to treat vascular complications associated with obesity. We have certain pharmacological agents, like currently available drugs that has been shown to activate AMPK, such as metformin, rosiglitazone, as well as aspirin. So these are commonly used in the clinical setting, but most of the studies using these drugs on AMPK concentrated more on the endothelium. So not much studies done on the pivot AMPK system. So it would be interesting if we can continue the study to look at the effects of this pharmacological therapy on AMPK activity in pivot in obese model. And drugs aside, there are also physiological activators or AMPK that we can apply, such as calorie restriction, fasting, and exercise. All these have been shown to activate AMPK, and this may partly explain the beneficial effects of these lifestyle modifications to improve vascular health.
Thank you.
Thank you, Prof. Umar. Is there any other question?
Dr. Aziza just now answered my questions. I was about to ask about the currently used drug which have MPK modulating activity, and you replied. So thank you.
Prof. Igor, apart from the drugs, there are also natural compounds that can activate AMPK, such as resveratrol. Yeah.
Okay.
But the results are limited to preclinical findings like in vitro and also animal studies, not so much clinical trial.
In your study, you didn't try to make the link with endothelial dysfunction or endothelial function. Are you going to study in the future?
Yes, Prof. Initially for this study, we want to establish the effect of PVET, AMPK on the anticontractile function as well. So that's why we purposely denuded the vessels from endothelium just to exclude any influence that the endothelium may have on the anticontractile effect. So in the future study, I mean, in the project, one of the projects that I'm working on now, the plan is to see whether there is any interaction between the endothelium and the PVET AMPK system in modulating the vascular function. Yeah, that's a good question. Thank you.
Thank you. Thank you.
Thank you, judges. And again, thank you, Dr. Aziza, for the presentation. We will now have 2 minutes for the judges to give their marks. Well done to all finalists. It was a fabulous contest, and I hope this session will encourage more young researchers among the MSPP members to participate in the next Young Investigator Award competition. I'd like to thank all the judges, contestants, and the organizing committee for an excellent job. A gentle reminder to all the judges to submit the marks to Dr. Hamid via email. And I know everyone is excited to know the result of this competition, but the final result will only be announced during the closing ceremony of MSPP scientific meeting. So make sure everyone will be there to know the results. Before we end this session, may I request judges, presenters, and organizing committee to switch on their camera for a photo session. Okay, I have the organizing committee, Dr. Alina, Dr. Shafi, to on the camera for photo session. I think Dr. Hamid will help us to take the photo. You can start with counting, maybe.
Okay, sorry. Okay.
Dr. Tan and Dr. Tan can on his camera. Dr. Alina probably
left. The
left.
host doesn't allow me to.
Really? Right now?
Okay. Okay, get ready. 1, 2, 3. Okay, another one. Okay, smile. Okay, 1, 2, 3. Okay.
Okay, thank you, Dr. Helmi. Okay, thank you, everyone, ladies and gentlemen. That wraps up our event today. On behalf of the organizing committee of MSPP Young Investigator Award 2021, I want to thank all of you for making your time in this in your busy schedule to join us here this afternoon. Let us end this session today with Surah Al-As and Tasbih Kifarah. I wish you all a pleasant day. Stay safe. Assalamualaikum and goodbye. Thank you, everyone.
Waalaikumsalam.
So those who are joining the MSPP AGM, I think the link has been sent by the secretariat, so you can join there. Thank you.